Hello, we'll continue our lectures about the organization of DNA into chromatin. By the beginning of the 21st century, molecular biologists had completed the sequence of the entire genome of hundreds of virus, bacteria, and one unicellular organism, eukaryotic, which is the body yeast S. vici. By now, genetic sequence is known for the fission yeast S. bombay, the simple plant A. thalania, rice, and the multiple multicellular animals, including the roundworm C. elegans, and the fruit fly Drosophila, mice, and human. Surprisingly, DNA sequencing revealing large portion of the genome do not encode messenger RNA, which is called non-coding DNA, and this constitutes about 98.5% of human DNA. So actually, it's 1.5% which is coding. All non-coding DNA collectively called junk DNA. This non-coding DNA can be summarized into introns, repetitive DNA, and regulatory sequence. The repetitive DNA are the telomeres, VNTR, variable number of tandem repeats, and transposon DNA. The length of the cellular DNA is a significant problem. The DNA is in a single human cell measures around 2 meters and must be contained within nuclei with a diameter of 10 micrometer. There is a specialized eukaryotic proteins, which is the histone, which associate with the nuclear DNA, fold and organize the DNA so it fits inside the nucleus. In eukaryotes, the DNA blastostones are called chromatin and can only be visualized as individual chromosomes during mitosis. The function of the chromatin is packaging DNA into smaller volume to fit in the cell, and as well maintains the integrity of DNA during cell division, and to provide as well access through an organizing fashion in such a way that specific DNA sequence within the chromatin are readily available for transcription, replication, repair, and recombination of DNA. So actually the condensation and decondensation of a chromatin will enable these processes, either transcription and replication, to occur. Once there is a condensation, the processes are stopped. The histones. The histone protein are 5 types from H1 to H4, rich in positive charge amino acids to interact with the negatively charged phosphate group. As you can see from the photo, the histones, the DNA is wrapped around the histones, separated by a linker DNA. The structure of the nucleosomes. A nucleosome consists of protein core of DNA wound around its surface. The core is octamer, 2 copies of each histone, 2A, 2B, 3, and 4. Wrapped DNA, the length of the wrapped DNA is around 147 base pair of DNA. Nucleosomes are linked by linker DNA, which are around 10 to 90 pairs. DNA of the nucleosome is less susceptible to nucleases than the linker DNA. That means that the nucleases can digest the linker DNA, not the DNA around the histones. If you look to the levels of condensation of the chromatin, there is a multiple of levels. Start from the beads on the string, then the 30 nanometer fiber, then loops of 30 nanometer fiber, then there is a higher order of chromatin folding which lead to the interphase chromosome. So that's the level of chromatin condensation. If you look closely, this is the naked DNA. The first level is the beads on a string, in which the DNA is wrapped around the nucleosome. This is followed by a higher order of magnification called the 30 nanometer fiber, in which these nucleosomes are again wrapped around itself to form a thicker structure, which is a 30 nanometer fiber. There is 2 proposed models for the structure of the 30 nanometer fiber, the solenoid model and 2-start helix model. The solenoid model is the widely accepted, while the other one is the strongly supported. The first figure shows the solenoid model, while the other figure shows the 2-start helix. Actually, you can see that both of them looks like a zigzag, but the other one, the 2-start helix model, it's a zigzag in different dimensions. Okay. This 30 nanometer fiber also include the H1 histone, which is the 5th major histone type. H1 is bound to the DNA as it enters and exits the nucleosome, okay, as a lock for the DNA that is wrapped around the nucleosomes. The other level, that the 30 nanometer fiber start to loop around to form a higher level of condensation. Now it's called loops of 30 nanometer fiber, with another higher order of chromatin folding and more folding leading to more condensation. During interphase, chromatin is dispersed through much of the nucleus. During cell division, folding and compaction of chromatin produce the visible metaphase chromosome. So chromatin exists in extended and condensed form. You can see here the figure shows region of the chromatin actively being transcribed and replicated because it's loose chromatin, it's not condensed. So here chromatin appear as extended beads on a string, while in this case the regions that are not being transcribed or replicated look like a more thick structure due to condensation, and here the chromatin appears as a condensed 30 nanometer fiber in diameter. What is the conservation of chromatin structure? The amino acid sequence of the histones are highly conserved. For example, the histone 3 for the sea urchin and calf thymus differ by only one amino acid, while the histone 3 of the garden bee and calf thymus differ only by 4 amino acids. The similarity in sequence among histones from all eukaryotes indicate that they fold into very similar three-dimensional conformations. Other minor histone variation exists. There is H2AX, which replaces H2A in a small fraction of nucleosomes in all regions of chromatin. And this H2AX participates in chromosome repair process at the site of if there is a double-stranded breaks. SNPA replaces H3 in the nucleosome at the centromeres, and this one participates in binding of the spindle microtubules during mitosis, while H3.3 replaces H3 in transcribed regions of DNA. In other words, the regions which are around 1.5% that is coding have the type of histones for H3, not H3 have H3.3 in the transcribed regions of DNA. What is a metaphase chromosome? The metaphase chromosome, actually it's a higher level of condensation leading to the decrease of length in the structure, generating the condensed form observing during metaphase. And it's on the only structure of the nuclear material that you can see it under the light microscope. Each chromosome contains just one linear DNA molecule. Condensins, a very important protein complex that composed of a structure maintenance of chromosomes, that's the proteins SMC, and this contributes to the chromosome condensation using energy from ATP hydrolysis. So what is the events in mammalian chromosome condensation? You see the first level is the 30 nanometer fiber chromatin. You have the second level around from 100 to 130 nanometer, which is called chromanema fiber. You have the third level, which is called the middle prophase chromatids. You notice that the thickness or the diameter of the fiber is increasing because there is a process of condensation. The middle prophase chromatids. You have the fourth level is the chromatids actually, which are bar-shaped structure observed only during mitosis, and then ultimately the two associated daughter chromosomes generated by DNA replication condense into chromatids that are linked by centromere. During metaphase, chromosome can be distinguished by banding technique, which is called karyotyping. By using a certain dye that selectively stains some region of the metaphase chromosomes more intensely than other regions, produce characteristic banding patterns that are specific for individual chromosome. This banding technique can help to distinguish chromosome of similar size and shape. This technique also uses probes specific for sites scattered along the length of each chromosome. The probes can be labeled with several different fluorescent dyes. So in other words, you can use a probe that is labeled either by radioactive or a fluorescent dye to detect specific regions in the chromosome. When a sample is observed with a fluorescent microscope, information is conveyed into a computer using a special program that assigns false color to the image and try to colorize the chromosomes. These computer graphics allow the two homologous of each chromosome to be placed next to each other and numbered according to their decreasing length. This chromosome painting technique is a powerful method for detecting chromosomal anomalies and in comparison and in comparing karyotypes of different species. So you can see here, this is a representative figure for karyotyping. So you can see the chromosomes are painted using the software, and then they are aligned each homologous chromosome together, and they number based on the length. So actually this is a karyotype of a male individual. And this is the technique which described you can take a blood sample, do centrifugation, and then put a hypotonic solution. The red blood cells, the white blood cells will, you'll get a white blood cells that with a due to the presence of hypotonic solution, the white blood cells will burst, releasing its DNA, putting a fixative, putting a stain, spread on a slide, and then observe under the microscope. So how we can detect by karyotyping a banding technique, the banding technique, the chromosomal translocation can be analyzed by chromosome painting. You can see here this is a type of a disease generated by the translocation of a part of chromosome 22 to chromosome 9, resulting in a chromosome 9 that have a part of 22 and vice versa. The small chromosome which is generated, which have a part of chromosome 9 is called DER or DER22 and usually named Philadelphia chromosomes. These characteristic chromosomal translocation are associated with certain genetic disorder. For example, this is the type of a chronic myelogenic leukemia contain the Philadelphia chromosomes. In other words, contain two other versions of the normal chromosome, which is the shortened chromosome 22, DER22, and they have abnormal long chromosome 9, DER9. And you can see under the microscope, the fluorescence show the normal chromosome and abnormal chromosome. The normal chromosome takes around takes the orange color, while the normal chromosome 22 takes the green color. In case there is translocation, you can see a mix between the green color and orange color. These forms result from a translocation as well, as I said, from between chromosome 9 and 22. So let's talk about the synthetic dogma. Synthetic dogma, actually, the DNA transcription, RNA translation produce a protein. The gene is a heredity unit of DNA that contain all the information to specify synthesis of functional RNA to build the proteins. Actually, the gene is read read by a 4-base language AGCAT that is through transcription, the language is changed to another language of RNA, which is AGCU, that upon translation, the language is 20 amino acid language of protein, actually, because protein generally made up of 20 amino acids. If you look closely into the structure of RNA, of course, you don't need to memorize the chemical structure. I'll go fast through this. The primary structure of RNA is very similar to that of DNA, with two exceptions. That ribose, of course, has a hydroxyl group at the 2' position, and T in DNA replace U. The presence of T rather than U in DNA is important because that provides stability for DNA. So why is OH in the RNA exist? Because OH is more chemically labile than DNA, so it has the affinity to do a lot of reaction, because of course RNA, once its function is done, it had to be degraded. So OH is the active group that can be used for degradation of RNA. As well, these OH provide a chemically reactive group that take part in RNA-mediated catalysis. RNA is cleaved into mononucleotide by alkaline solution in order to be recyclation, and the ribonucleotide can be used again for the making of new RNAs. Like DNA, RNA is a long polymer, consists of and have 3 major types: messenger and transfer and ribosomal RNA. And the RNA can form a hybrid helix composed of RNA and DNA strand, or RNA double strand, or RNA double strand. Always have the conformation of A, which differs from the B form of the DNA. Some RNA molecules have a catalytic capacity. Ribozymes, they catalyze the self-splicing once the function is done during the formation of the majority of functional messenger RNA. In addition, of course, of messenger RNA and the 3 major types, there are thousands of additional genes that are transcribed into non-protein coding RNA, like small nuclear RNAs, small nucleolar RNAs, and large families, around 2,000 in human, of short microRNAs. Unlike DNA, RNA are single-stranded and exhibit a variety of conformation. So what is the conformation of the RNA? Actually, the primary structure is the linear form of the RNA, while the secondary structure, the first secondary structure is the hairpin. You can see when a sequence that is complementary over 5 to 10 ribonucleotide, it can base pair to form a hairpin. While if the number of nucleotide is more than 11 to 100, it can form a loop with a stem with the complementary bases, and this is called the stem loop configuration. So basically these two structures are considered to be a secondary structure. You can see a tertiary structure, and actually it's a combination between two stems and two loops, and we call it here a pseudoknot. Actually, why pseudoknot? Because it's a fake knot, because once the RNA is ready for doing its job, it had to be melt to be linear. Transcription of messenger RNA. So you can see here the template of DNA and the started transcription of RNA from the direction 5' to 3', and these ribonucleotides start to be added in this direction and synthesized by the RNA polymerase. Once there is an incoming ribonucleotides, like for instance ATP, GTP, ATP, UTP, CTP, GTP. Once there is incoming nucleotide, it starts to base pair complementary with the template DNA, and it starts the OH with the alpha phosphate. There is a process of polymerization where OH react with the alpha phosphate. This forming the high energy bond between this the high energy bond between alpha and beta phosphate of the ribonucleotide is replaced by a lower energy bond between the OH and the alpha phosphate. The result byproduct is a pyrophosphate. With the help of the pyrophosphatase is converted into a phosphate molecule that can be used to build new nucleotides. If you look closely to the DNA structure above, so you can see +1. So some numbers is important to understand here. +1 is the position of the start of the gene, whereas the coding sequence is downstream +1, while the numbers that have a minus is upstream of the gene. And actually the region that before the gene is normally called the promoter sequence or promoter region. So actually transcription, the coding sequence starts from +1. So you can see downstream denotes the direction in which the template DNA strand is transcribed, and again the direction of the movement of the RNA polymerase over the DNA. So RNA polymerase moves downstream of the +1 position. Upstream denotes the opposite direction. Nucleotides in the DNA sequence downstream from the start site are indicated by the plus sign, while the upstream are by indicated by the negative sign. Because RNA is synthesized in the 5'3' direction, RNA polymerase moves down the DNA strand in the other direction. So you can see here, again, the DNA is converted into sequence, into non-template and template strand. The template strand, you can see that it is transcribed into complementary to the template strand. Of course, U is in place of T. So we'll talk here about transcription of messenger structure. The transcription of messenger RNA generally is divided into 3 major steps: initiation, elongation, and termination. So we'll start talking by the prokaryotic transcription. So let's start with the initiation phase of the prokaryotic transcription. At the beginning, the DNA is closed. The green spot is the start site, the red spot is the stop site, and of course you have upstream of the start site, there is a promoter region. The RNA polymerase start to require initiation factors, IF, to recognize a promoter. This RNA polymerase, once it recognizes promoter, it starts to be ready. And this structure is called closed complex. Why closed complex? Because the DNA is still closed. Once the RNA polymerase recognizes promoter, it starts melting. The RNA polymerase of the prokaryotes have a helicase activity that melts DNA. The melting of DNA converts the DNA structures, the complex, from a closed complex into an open complex. And the transcription bubble is formed which have the size of 12 to 14 base pair. So the melting happened by the help of the RNA polymerase. Once the melting happened, the RNA polymerase started to transcribe the first nucleotides. Once the first two nucleotides have been transcribed, that's meaning it is the end of the initiation phase. So initiation phase is completed. Okay, so you have two after formation of or transcription of the two nucleotides. So we'll talk about the elongation. Actually, the elongation, it is a repetition of what have been done before. So the RNA polymerase starts to move over the DNA, as well as the transcription bubble is converted to a melted region around 14 to 20 base pair and start to move along the DNA. And you can see that the nascent RNA start to be emerging with the 5' end free. That's meaning that the transcription is occurring in the direction of 5', 3'. The RNA polymerase adds a nucleotide, one ribonucleotide at a time, added at the 3 end of the nascent RNA. It's important to note that usually it is always the RNA is bound to the DNA for the last 8 nucleotide, and this is the hybrid region of DNA RNA. And if you remember, we have said in a previous style, the structure of a DNA RNA hybrid is a A form, not B form. And as long as elongation proceeding, it moves adding one nucleotide at a time and preserve the melted region to be from 14 to 20 nucleotides. For determination, once the RNA polymerase reach the end of the gene, the red spot, all are released. The completed RNA strand, the nascent RNA strand, and RNA polymerase is released for another round of transcription. RNA polymerase can transcribe very long genes to around 2 million base pair without dissociating. This means it's very stable complex from the DNA or releasing the nascent DNA. It's important, this information is very important. RNA synthesis occurs at a rate of about 1,000 to 2,000 ribonucleotides per minute at a 37 degrees centigrade. So actually the average that you need to study is 1,500 ribonucleotides per minute. Thank you.